take questions if you like as we go. But it's lovely to meet you all. Hopefully get a kind of sense of what you're all doing by the end of this as well through the questions. But purpose of today, Brona asked me to speak to you about farmyard manure and compost and things that you could do perhaps to get a little bit more out of it, and also to tie it in with the biological side of things, which is what we're predominantly focused on. So the idea of this is really to sort of enable you guys to understand a little bit more about compost, farmyard manure, and maybe even composting farmyard manure, and some of the factors that might influence kind of a bit, help you yield better performance, better kind of use of those certain materials as well. And hopefully you'll learn a little bit about soil biology as well. So we've got quite a lot to cover. I think it'd probably be good to try and end with a Q&A at the end of the session. I think me talking for 2 hours is probably enough for anybody. I've got a pint of water though, so I'll try my best to kind of keep up. And similarly, if the pace is too fast or too slow, do also let me know as well. Kind of, Brona, I think you're the only one I can see, so you need to kind of wave at me. So yeah, this is what we're going to cover. I'll talk a little bit about me, Soil Biolab, what we do, and come back to that a little bit later on as well. I'm going to have a brief kind of overview of the differences between FYM and compost. Then we'll talk a little bit about colloidal solids, then managing nitrogen, and kind of in unison with that, just touch on carbon nitrogen ratios, which are absolutely fundamental to so many things in the soil world. It's unbelievable. And we'll talk a little bit about composting theory of a particular type of composting. Maybe a little bit about the different types of compost, and then extracts, which is the other thing that Brona wanted me to talk about as well, because I understand a few of you are either looking to make your own extracts or kind of biostimulants, or already doing it. So we'll cover that, and then we'll kind of wash up at the end, if that's all right. So a little bit about me. I founded Soil Biolab just over 11 years ago. I used to work for another laboratory on a big organic biodynamic farm in Hampshire. And before that, I spent over a decade in horticulture. So we've always been kind of used to being around people who grow things of sorts. One of the reasons we set it up was, and I say we, I've got a silent business partner in Germany. But one of the reasons that we set it up was really to try and be an independent source of information for people, not kind of focused on selling products, which over a decade ago, a lot of the kind of bio companies were educating in order to sell to people. We wanted to be an independent that would help people analyze certain things, primarily their soil, compost, maybe even products that are being put their way, and give kind of an impartial assessment of those things. So we are staffed by various people. We're a very small business. There's 4 of us in total. Got a couple of soil scientists that work on the day-to-day for me. We have kind of soil science as a background, or plant science as a background. We've got global links. So we've always had an association with a company called Earthfort in Corvallis in the States. The guy who runs that was one of the kind of founding supports for Elaine Ingham for the Soil Food Web. He helped her kind of launch the laboratories across the globe back in the day. So we've always had kind of their support, which has been fantastic. And now we deal with all sorts of clients, ranging from farmers and growers to agronomists, arboriculturalists, greenkeepers, anyone and everyone in between. And we've always kind of been driven to provide the highest quality expertise and advice that we possibly can. So this is our kind of show off slide, our wall of fame. There's more to add to it. Some of the most notable ones, early days Soil Association, their innovative farmer kind of hat on. We were involved with them doing large scale arable trials of compost tea, which was really interesting. I've always helped organisations like Organic Research Centre doing their trials, and actually we're just coming to an end of a piece of work that they've been doing on different compost as it happens. And then we deal with kind of big sort of landholders like National Trust and the RSPB. Last 2-3 years been involved working with those guys, helping with the major oak up in Sherwood Forest, the Robin Hood oak, which is pretty cool. So it's quite a diverse array of people that we deal with. I suppose agriculture is kind of our bread and butter. It's where we started, although right in the beginning, it was probably the smallest client sector that we dealt with. Not very many farmers would send us soils. Most of it tended to come from high value crops, so kind of viticulture, protected ornamental crops, things like that. So it's nice that it's now kind of swapped around, and we're helping a lot more farmers kind of manage their soils for better plants, crops, animals, and all sorts. So that's kind of background about us. This is what you came for. Farmyard, manure and compost. So we're just going to touch very quickly on the key differences. And I'm sure, as you all know, farmyard manure, it's raw animal waste, but one of the primary benefits of it is that quick supply of nutrients, kind of a large nutrient value. You get kind of other benefits such as the organic matter that comes with it. Even humates, often sort of seen as that brown liquid leaching out of big piles of manure. Some of the downsides, though, relate more to the hygiene side of things. So this is a fantastic close-up picture of E. coli, especially when you're dealing with cattle manure. This is one of the primary kind of hygiene concerns to be aware of. It does come with its kind of health-related risks. So handling of it's got to be considered, and we'll touch on that again a little bit later. Moisture as well is incredibly difficult to manage, but all of this is in comparison to compost. I think this is a lovely kind of storybook image of composting, but actually I did, after putting this in, notice a couple of things that were wrong. One of them is the uncomposted vegetables that are sat in it, and the other is the broken pallets at the back. If they'd done it right, wouldn't be spilling all over the floor. But compost, whether it's compost, or composted manure, this is the act of decomposing organic matter through microbial action. In doing that process, you have an improved nutrient availability, and you can reduce some of those nasty pathogens that I mentioned. And then you can put it on your lovely trailer on the back of your shiny tractor and spread it. It can help you build up soil organic matter, and composting enhances the nutrient content of the material, the original starting materials. Fundamentally, it transforms those nutrients present, whether it's in manure or your kind of green and brown materials that you've collected, and it enhances their availability and stability for crops. While raw manure does give those kind of nutrients, the act of composting itself makes them even more accessible and balanced for plant uptake, and a bit more stable as well, which we'll come on to shortly. Can I just check, is the pace of this all right? Yeah, thank you. This is a lovely picture. I'm sure many of you will have seen this already. This is well, one of the original kind of pictures of the soil food web. And you note that I'm kind of erring slightly towards compost a bit more than manure here. A lot of these benefits might actually ultimately come through manure that's been set aside and left for some while. It will continue to break down, but primarily I'm kind of focusing on compost here because over the years I've been asked, is it really worth composting my manure? Should I be thinking about it? And generally the answer is yes, but And there's a whole heap of reasons why you might not: practicality, cost, um, machinery, the resources. Sometimes you just need to kind of move things from a barn to a pad to a field as quickly as possible. Other times you might be kind of storing things away. But in any event, there is microbial decomposition happening there. And when you have a composted material, or a well composted material, which is carried out by bacteria and fungi, those guys will carry out a process called mineralization. So that is basically converting the organic nutrients in those raw materials into inorganic plant soluble forms. So, and then those mineralized nutrients become available for further processing by things like your protozoa and nematodes into highly available forms that then go into the soil solution, and your plant and crop roots can uptake quite readily. So even when we're talking about the principles of compost and some of the benefits out of that, we're actually touching on a lot of the principles in soil about that succession, bacteria and fungi breaking things down, and then bigger creepy crawlies, second trophic layer protozoa are going to feed on those bacteria and access those nutrients held by those bacteria, and then things will come along, earthworms, eat the protozoa and move them up the chain as well. So incredibly important. That's is important because later on, when we talk about carbon and nitrogen, all these things are kind of interlinked. It's quite confusing at times, but I'll try and make it simple as I can. So kind of in summary, when we look at the differences between compost and manure, there are lots of pros and cons. I suppose the Farmyard manure is a great fertilizer primarily. It will add organic matter. There are some downsides associated with that. So the nutrient availability could be a bit variable. It could be very wet and heavy to work with. You are often applying anaerobic microorganisms to a soil where, when it's working well, it tends to be an aerobic environment. So that can have implications. But I think it's fair to say, as long as you're not applying FYM in excessive volume all the time, everything in moderation, it shouldn't be too much of an issue. But all of that's kind of compared to compost or composted manure, where you don't have issues such as the same kind of strong smells. It might be slightly lighter and easier to distribute, whether that's through a spreader or even kind of hand spreading, quite frankly. The volume is reduced, and then you've got kind of things like the environmental impact, lower emissions, reduced leaching, and probably structurally a compost is going to have slightly better or slightly more long-term benefits to your soil. Does that all make sense? Fab. So I said I was going to talk a little bit about colloidal solids. These are the tiny particles that remain in a suspended kind of mixture of soil or compost. This is what they look like up close. Another great excuse to show you a picture from an electron microscope. This is not, by the way, the kind of level that we zoom in on. All of our work is done by human eye, kind of in the visible, up to 1,000 times magnification compared to this, which is infinitely smaller. Colloidal solids are really important, and I kind of mentioned that was trying to, through this presentation, give you ideas and tips for things that perhaps you're not doing that you might want to think about doing or exploring to improve what you're already producing. And the addition of colloidal solids, aka soil or compost, to your manure or compost or composted manure can have massive benefits. So yeah, you heard me right. Adding compost back to compost can be a good thing. You'll notice from the structure, it kind of looks like a meteorite or don't know, like a fragment of the moon almost. There's all kinds of nooks and crannies. Often it's referred to as a complex colloidal matter. Um, so because of all these kind of little surfaces and nooks and crannies to tuck things, colloidal matter has, or colloidal humus, I should say, has a very high cation exchange capacity. So that means the ability to hold on to nutrients. It's negatively charged, so it means it attracts positively charged ions. So that would be the cations, things like ammonia, ammonium, potassium, calcium, magnesium. It will tightly bind them on its surface. Also, it provides an excellent house for microbiology. So it's a great food source. It's a great habitat for all the kinds of microorganisms, the bacteria, fungi, even protozoa, nematodes might swim around through them. But it's a great resource for those to utilize. And the addition of it within your compost during the composting process would kind of magnify, if you like, the end result of your compost. You would end up with what is often referred to as humified compost. So that is kind of one of the benefits. The other relates more to kind of the structure of the material you add it to. So again, all those little nooks and crannies, it provides an excellent space for retaining water, and it can help retain somewhere between 10 and 20 times its own weight in water. And that can be a real benefit for all sorts of reasons. Water is a key driver behind biological activity. We know from various tests that we've done over the years that it's probably one of the single biggest influencing factor, certainly across the seasons, in terms of how biologically active a soil can be. And in fact, we're coming up to July tends to be one of the kind of driest, certainly over the last 7 years, it's been one of the driest months, and we record the lowest biological activity. Weirdly, excuse me, weirdly, December is one of the most active. And I can tell you now, 10 years ago, when we started testing people's soils, we used to have things called seasons where, November, December, January, February, they were cold, soils were cold, microbes were dormant, and we were praying for spring kind of by the end of February, early March to start to have things to do again. And then you'd move through a nice hot summer and end up with kind of a mild move into autumn before things started to cool down, and you'd end up with this kind of pattern across the year of biological activity. And then somewhere along the line, seasons have turned topsy-turvy. We get incredible extremes in the weather, and really weird things have started to happen, like soils seemingly become active all year round pretty much. Used to tell clients that after 4 or 5 consecutive hard frosts, that was the time to stop sampling your soil, because you wouldn't get as much value out of the test results on your soil microbe analysis that we provided. You just wouldn't get the same value out of it. But certainly over the last 2 or 3 years, I can't remember when we've had a run in the UK of more than 3 hard frosts on the bounce. So, but moisture content as well as temperature, which I kind of touched on there, is a huge factor in how active your bacteria, fungi, your protozoa, nematodes will be in your soils, in your compost, or in your manures. So anything you can do to help buffer that and control or regulate, so much the better. And that means when it rains too much, your compost is able to absorb a little bit more than perhaps just the bare soil, or when it's hotter and drier and water is a bit scarcer, there's a little bit more reserves there to be drawn from. And then the other kind of benefit, I touched on it just then, really is about this home for microbes. So it serves as not just the primary energy source, but a physical substrate for things to kind of live in. And then they can drive that mineralization or phosphorus solubilization, as well as the organic matter decomposition. So what you're looking at here is it's a bit of a grainy picture, actually. Sorry about that, but that's fungal strands crawling through the soil. So fungi are multicellular organisms that kind of prey on more complex carbon materials in the soil. So they will be kind of swamping that and beginning to break it down. They're what's called saprophytic fungi. I'm just going to have a little drink, 'cause I'm drying up already, all this talk of water. Okay. So we're going to touch a little bit on nitrogen management before we move into carbon and nitrogen ratios for a bit. But again, this is kind of structured in a way to try and give you kind of actionable tips, or at least takeaway principles that you can kind of think about and maybe it'll help you alter what you're doing. And one of the first things that I kind of thought of was about how to control the loss of the nutrients, which often, not always, and point of this really is also to kind of instill about the importance of microbiology. But let's kind of jump back a few years. The primary benefits of manure and compost was as a kind of source of nutrition, and especially with FYM, nitrogen being one of the key kind of benefits from it. And if you just stacked your manure and left it, you will lose a lot of the benefit of that if you do nothing to it. Similarly with compost, if you just kind of build it and leave it, if you're lucky, after a long enough period of time, things might have broken down and produced something that's usable, but quite often you can lose a lot of the nutritional benefits of the original materials that you started to, and just could have captured that much more. And that's to do with aeration. So proper aeration supports the activity of the nitrifying bacteria, which are responsible for converting the ammonium into nitrate, which is the plant soluble form that you kind of want. Now, if you've got sufficient oxygen, that prevents excessive ammonia being lost through volatilization. That's basically where the gas escapes into the atmosphere. Similarly, if you've got kind of a hot, very alkaline type of environment, which I know from commercial, let's say commercial kind of large industrial scale composting. I used to run an 80,000 ton composting site. That was that was big and scary. Whenever a machine broke down, it ran into the thousands straight away. Even when everything was working well, it would still cost you a couple of £1,000 a day in diesel and hammers for the shredder. So yeah, big and scary. But anyway, that type of scale of composting tends to produce very alkaline type of compost. So pH is kind of well north of 7, maybe not quite 9, but definitely low to mid 8s. Anyway, where you get those hot alkaline conditions, that can kind of promote that loss, whereas cooler, lower pH type environments promotes the formation of ammonium, which is much more stable. It's kind of why the holy grail of composting is low pH, but when you're talking certainly thermophilic compost or composting, that's quite a hard thing to do if you're not managing it correctly. So yeah, anaerobic, low oxygen conditions, often caused by insufficient aeration, can lead to this denitrification process as well, where things kind of revert back and you end up losing more nitrogen through gas from the pile. And you can kind of tell these things from simply by the smell using your nose. What you're looking at here is a tractor drawn compost turner. And by the way, I'm not going to tell you how to make compost through this, but I was taught by a very clever Dutch guy who did lots of composting, lots of people across the world, that the optimum size for your windrow, this type of compost, is 3 meters wide by 2 meters tall. And a tractor drawn turner would be more than sufficient to turn through all of that. There's actually a whole heap of science that sits behind it that is probably around 60, 70 years old that kind of validates that. And I've yet to see any modern study that comes anywhere close to that. So, yeah, I'm a big advocate for thermophilic compost, so kind of windrow or composting through kind of using digestion, heat, and microbes. I'm not anti others. It's just I'm particularly keen on that kind of method. So yeah. Anyway, moving on. The other area where people often fail is through not managing moisture. Now, there's actually kind of a wide range of views people have on this. When building compost piles or rows, the ranges tend to be somewhere between 40 to 50%, but can go as high as 60%. The challenge at the upper end is too much moisture saturates particles, you inhibit oxygen flow, you can lose heat, which is quite important, and then you get these anaerobic zones or pockets throughout the batch that you're creating, and then that whole kind of gassing off effect occurs whenever things are smelly. That's gases being formed that you want to retain in the material in your pile. So a simple way to check, you don't necessarily need complex material, but it's the squeeze test. That's to say, you kind of get a load of your compost. I suppose you could do this with manure if you are brave enough as well, but kind of squeeze it and it should feel like a wrung out sponge, they say. Perhaps a more accurate way that occurs to me is you could oven dry this yourself as well. So take a cylinder of a known volume, weigh it, fill it with compost. You could do this for soil as well as a more accurate measure of soil moisture content. Put it in an oven overnight. You can run it at a fairly low temperature, kind of 100 degrees or less, and then reweigh it. You can keep doing that until it stops reducing in weight, but usually it's kind of overnight. Reweigh it, subtract the weight of the container that originally held it, and the difference will be the moisture, which you can calculate as part of the volume of soil compost originally taken. You can get your percentage kind of weight loss in that regard. So that's good thing to do. The other thing is, and I'm back to kind of 3 meter by 2 meter tall windrows again, but this can apply to manure heaps, it can apply to static piles, it could apply to any form of compost that you've even made and stacked up, but it's covering it. Obviously, if it's an active compost pile or manure pile, you want a breathable material because it still needs to get oxygen into your microbes that are doing the digestion process. These particular blankets that you see are a material called Top-Tex. It's incredibly expensive. Top-Tex, the brand, it's a bit like Gore-Tex, I suppose. It's someone's probably got the IP on it somewhere and is making an absolute mint out of selling the stuff on, which most of it I think tends to come from Austria. But the beauty of it is it protects the material from rain, so it helps you kind of manage your moisture content. It keeps the heat in, and when you stack material up, unless it's kind of bunded or in a clamp, it's exposed to wind, and that wind can drive through and kill off the composting process. But these ones are breathable. It allows kind of that gas exchange in and out as well. So yeah, temperature, gas exchange, as well as protection from wind and water, kind of crucial. So you don't need to spend a fortune on these kind of Gore-Tex blankets of the composting world, though. I suppose at an absolute worst, dare I say it, a tarp, but be mindful of the fact that you are reducing the amount of oxygen that's available. If you've got finished material, it's probably better to try and store it under cover altogether. If you're looking to set aside materials for an extended period of time, if you can, put them under a tree. That was another thing I was taught as well. One of the reasons, as long as they're on soil under a tree, that soil underneath it will inoculate your your compost, your manure, as it's maturing and sitting there. So you'll end up with something that is even more biologically active than when it finished. The last kind of principle on making things, which I'm not going to go into massive detail on, is just about layering materials. And it's my understanding that even if you're involved in something like vermicomposting, so someone's ears prick up them, it is important to layer the materials as you're kind of building your cake. Ultimately, one of the reasons for this is to kind of homogenize the material, and you see here that things are kind of laid out as green and brown, which we're about to kind of move on to, carbon and nitrogen. But really, this is about homogenizing material, making it as easy as possible to kind of blend the material so it's as uniform and consistent as possible. And actually, here's a tip: if you're ever sampling manure composts to send them to a lab, it doesn't have to be us, it could be other labs, they do exist. Sampling your batch with the same kind of mindset as you would your soil, that's to say, to get something as representative as possible. That means sampling in different positions along, different heights, and also different depths within the material too. And if you've done it correctly, everything will be mixed through and it will kind of be the same near enough throughout. Maybe not quite on the outer where things are exposed to light and the elements, but just beneath the surface, everything in theory should be quite similar. Cool. So we're going to talk a little bit about carbon nitrogen ratios, balances. Now, carbon nitrogen ratio, and these principles apply to kind of just straight manure, which is obviously going to have a very low carbon nitrogen ratio. It's one of the most vital elements for composting efficiency. The ratio dictates the balance of nutrients available for microbial decomposition. Again, dependent on where you look, you will hear different people cite what the optimum is for compost. That could be anything from 20 to 1. So that means 20 parts carbon to 1 part nitrogen. And when we say parts, I guess in practical terms, if you're building compost, we're talking by volume. So that could be bucket loads or spade loads. So 20 spades to 20 spades of, I don't know, wood chip to 1 of manure, for example. That could be deemed optimum in some situations. And then you hear other people say it could be as high as 30 to 1. There's even a camp that said you could go up to 40 to 1. The microbes that I've kind of mentioned, so those bacteria, fungi initially, and latterly the protozoa and nematodes that come along later in the process, and the same that are in the soil, they tend to have a carbon nitrogen ratio of somewhere between 5:1 and 10:1. And that's a whole topic of its own about nutrient cycling in soils and why microbes are really important. Having lots of soil life is key for that natural nutrient cycling. But these guys involved in the composting process, it's important as well because they're contributing to the carbon nitrogen balance of the mixture. The higher carbon element, so when we talk 20 or 25 carbon to 1 nitrogen, the reason you need such a high proportion of carbon is because that material is metabolized by the microbes, released as carbon dioxide, which is why often you see people with their kind of CO2 meters stabbing compost piles, making sure it's all working. And as a consequence of that, you lose the sheer bulk of material. It could be anywhere from 30% to 50%. You can lose that volume of material off as gas through just the process of composting. So you need a lot of carbon in the first instance. If you have too low a ratio, so something below 20:1, that means that there is maybe too much nitrogen relative to carbon. And that means that you could end up losing a lot of nitrogen again as a volatile ammonia gas. Again, you probably would smell that. If that's the case, the thing to do is to add more carbon-rich materials. So this is your kind of wood chip or clean cardboard, things like that, straw. Add that to readdress the balance. If you have too high carbon nitrogen ratio, so we're talking kind of way off the spectrum now, something like 40:1, say 40 shovel loads of wood chip with 1 shovel load of manure, that excess of carbon can lead to nitrogen immobilization. Basically, you just kill off the composting process. And this is similar to things like adding wood chip to soil, which is another question asked quite a lot. It's kind of one of the reasons soil has its own carbon nitrogen ratios and an optimal balance. If you blend and mix that wood chip in, you're giving it too much carbon relative to nitrogen, and so it will take before the microbes will take that nitrogen from the soil before giving anything back. So it's a little bit like Goldilocks's porridge. You need just the right amount, and different materials have different optimal amounts. This is a few. Years ago, I put together an Excel spreadsheet, which is much more detailed than this. Oh, I can see a question come up then. I saw someone has made the mistake of looking and saw a hand pop up. I'm happy to share that because I even tried to work out a way where you could enter volumes of things to try and help you work out the right mixture, and then you could start to layer things if that was your particular style of composting. At least it serves as a starting point anyway. But what is interesting, kind of the two on the bottom, you've got all the different materials that you might want to include in composting. These are just a few, mainly kind of manure led. In fact, I've got a quick question. What is the number one, or what animal I should say, does the alleged, because I've never looked it up, but I remember being told this by a reputable source, what is the best manure in terms of nitrogen value from what animal? In fact, we'll leave that as a question. You can answers answers in the chat, and we'll check in at the end. Yeah, it's surprising. Anyway, the interesting kind of ones highlighted is just this ideal soil. So we've got the kind of the composting one. That's the 25-1 is probably a safe bet to initially aim for. However, different materials have different carbon nitrogen ratios. Your horse manure might have a slightly different ratio to what is shown on this spreadsheet. The only way to know is to test it before you start. The ideal so we talked about the ideal kind of composting. Ultimately, that process you'll probably end up with something that's perhaps somewhere between 10 to 15 to 1 carbon nitrogen ratio as a finished product, perhaps. The ideal carbon nitrogen ratio for soil is 24-1. When I say the ideal, I mean, again, it depends on so many different factors. This is a sweeping generalization, but in our experience, C and N ratio 24-1 enables microbes, these kind of saprophytic fungi, the bacteria, means that the protozoa, the nematodes are all working optimally in that particular environment. But the key message from all of this kind of recipe building is the management of it needs to move beyond just recipe building, kind of putting your mixture together, putting in the oven to bake. It's an active process. It needs actively managing. That means using equipment like thermometers, moisture meters, or doing that moisture test I mentioned, checking it with your sight, your nose, turning it, watering it if you need to, adding bulking agents if you think that you're getting that nitrogen kind of lost through the smell, managing the size, keeping it out of the wind, all of these things. It kind of requires an integrated management approach to get the most out of it if you're kind of looking to invest in getting more out of your compost. So it's kind of a bit of a science, a bit of an art. You can take all the kind of textbook theory, but when you come to apply it, you need to kind of understand some of these principles so that you can adapt a little bit when it goes off piece.
Simon, I see John McElroy has a question here. Do you want to take it before we go to the next slide?
Yeah, go for it.
John, have you finished changing nappies? Do you want to? You ready to answer the question?
I have. I've got 2 kids that should be in bed, but anyhow, as well. Simon, thanks for this. Question: Is separated slurry solids a suitable material for composting, and if so, what's the C10 ratio of that? Ooh. And even digestate solids, even more relevant to myself.
Yeah. You can pretty much, and have been involved, and well, yes is the short answer. Second half, I don't know without looking it up, because it's been too long in terms of what the CNN ratio is and what you'd want to mix it with. I guess with digestates, the thing to be mindful of is kind of the accumulated concentrations of certain things. And if you're looking to incorporate it on an ongoing basis, obviously those things might kind of ultimately accumulate in your compost, and then when you apply it on your land, you start to build up concentrations there as well. But you can pretty much compost anything, and people have pretty much composted anything, and only regulation, the Environment Agency kind of put pay to where the environment I worked at and ran a composting site where you couldn't unfortunately dispose of carcasses of animals into it because of kind of hygiene. But if you are composting in a controlled way correctly, you can pretty much sterilize all of those harmful pathogens through the digestion process. So certainly things like certain chemicals. So goodness me, what was the? I remember in a minute. No, not neonicotinoids, but there was one, wasn't there, within the novamectins? Was that it in lawn care? Big no-no for composting, because you would just accumulate that in your end product. Similarly, heavy metals you'll just accumulate in your compost as well. But if it's all right with you, John, I'll see if I can find that information and include it on the spreadsheet, which I'll share with Brona, and she can share with you guys if you like.
Yeah, thanks, Simon.
Cool.
I have a follow-on question, Simon. Maybe you want to move on. Phosphorus is becoming it's already an issue, but it's going to become more prevalent with the new nitrogen action plan. Is compost a way of getting rid of it if you do have a separator? Is that, or is on a big scale operation like the one you were working on, is that not a viable solution for the pathogens and whatnot?
Sorry, I'm not clear what the question was. So phosphorus.
So we're gonna have too much P if we separate our solids.
Okay.
Is composting a viable way to get rid of that P? Is that, or is it not used by industry because of the reasons you're talking about? And is it not really a viable idea or option to compost separated solids?
Yeah, to try and reduce the concentrations. I suppose you could dilute it. I just wonder in terms of what volumes you'd be putting in, as to what how diluted it would be at the other end. Yeah, it's probably worth a trial, and then get a thorough analysis.
Can I ask Simon if most of us are dealing with farmyard manure in a large pile, and turning doesn't really happen, and we do just generally leave it for a year? I think the sample I sent you is 2 years old. Is there anything that we could be thinking about? Maybe is the height of it something that we could kind of look at as a better ways of managing it?
I forewarned that the dog would interrupt. Reduce the height, did you say?
Yeah, I know. I don't know how what everybody's would be. I would stack up to about 4 meters.
Right.
As high as I can get the arm to drop it.
I mean, you could do, but I would look to add other materials to it, perhaps. So I mentioned, I mean, okay, admittedly, not everybody has access to kind of large volumes of compost that you'd need to mix with a heap that big.
So say it was something like old waste hay or wood chips or something like that. Would you mean
wood chips? Yeah.
Your layers, just chuck on something like that would keep it very open before your next layer of dung.
Keep it open. It absorb any excess. You know, in essence, you move the C&N ratio, stabilize it a little bit more. And if you're leaving it there for kind of a year or more, they will continue. There will be some further decomposition take place. And if you're going to spread it on the surface, you don't have the same issues that I mentioned in terms of combining it with the soil and then kind of skewing that 24:1 ratio, optimal ratio.
Okay. Thank you. I think Johnny's got a question there. Do you want to go ahead, Johnny?
Yeah, thanks very much. I was very interested by what you said about the dead animals, and I thought it's just a comment to start. I thought that's a far better way of dealing with that issue than incineration.
Yeah.
And to me, incineration is a complete waste.
Yeah, absolutely.
Yeah. No, I think that'd be great if you could.
And I think it's to me it feels kind of I mean, especially for the squeamish out there, it probably doesn't sound particularly nice, but it feels a more holistic way to manage it. Do you know what I mean? Keep things on farm. Yeah.
Well, I mean, at the end of the day, I mean, if you had a carcass in the wild, it's obviously something that eats at it, but the rest of it rots into the ground. So
yeah.
And there's a lot more than just NP and K in a carcass, obviously. So
yeah, absolutely.
But the other thing I was going to say, sort of following on from what Bruno was saying about a big stack of manure in a shed. Say if you had two sides of a shed, and you stacked, say you cleaned out all your sheds and you put it on one side. If you just basically shifted all that material to the other side and kept doing that, is that sufficient turning until such stages that it's, I don't know, it stops steaming or something like that? Is that a fairly crude way of looking at it?
Yeah, whenever I've spoken to kind of farmers about composting, there's the really enthusiastic camp that understands the value of it and then tries to make it themselves and then realizes they can't get enough, and there physically isn't enough material on the farm. They don't generate the wastes to produce the volume that they would need. And then there's sort of the camp that would as soon as buy in, and that's absolutely fine. And in those situations, something's better than nothing, even if you end up resorting to kind of industrial made. We've got PAS 100 compost. Similarly, there is a science as well as an art to composting, but it's not people's day job. It's another activity, and don't get me wrong, it yields reward, but you've only got so much time, so much kit, so much that you could financially invest in, and so there are limits to things. So in terms of, I guess what we're talking about is kind of bang for buck. Yes, that would be better than not turning it at all. That will improve the quality.
And for instance, we have a lot of well, not a lot. We have a couple of, say, trailer loads of waste hay. And if you're going to mix that in with manure, are you better instead of tipping the hay on one bit and then tipping the manure over it, you'd be better trying to mix that and incorporate it more evenly.
Ideally, you would, kind of layer it, and then once you start to, every so often just shuttle it over again. You're rolling it and it's mixing it a little bit more. Yeah,
that's cool. Thank you very much.
That's all right. I don't know if that was one more question or the same hand that was up from John. Nope.
Oh, sorry, that's my hand from before.
Yes.
And take it down.
Cool. And we'll move on and just look briefly at kind of what's happening in that compost process. This looks a little bit grainy, so sorry if it's a bit difficult to read. There's different stages, and again, I'm referring to thermophilic process by and large, because if you are leaving manure to a degree, this is what's happening, albeit the ratios are different to a compost heap or row that you have built. But the same is applicable if you are composting your manures as well. It's a heat orientated process, and there's a few key stages. So the first is this mesophilic phase where you've got the initial breakdown, and these are kind of lower These microbes operate at a slightly lower temperature range, so 20 to 45 degrees centigrade. And they're beginning to break down the carbon material. And then the temperature will spike, and any books that you read or guidance, this is kind of the period where people are saying it's really important to let it get to a hot temperature. Now, again, at the industrial scale, this kind of graph shows you somewhere between 60 and 70 degrees centigrade. At an industrial scale, I think we were kind of going up to 85 degrees, not far off, not far off pasteurizing the material, to be honest. And that is purely, and kind of taking it there and holding it there purely so that you're killing off weed seeds and those pathogens. But in reality, if you're managing this process well, you don't need to go as high as that. And this kind of particular phase within the compost cycle, which is kind of really the key bit, is the thermophilic phase. So they are thermophilic, primarily bacteria that are breaking down the more complex materials, and there's all this heat that's kind of being generated. Because that heat's being generated, that is where it's really key to make sure that the compost can breathe. Or if you've combined your manures with other material just to try and hold on to more of that kind of nutrients, again, this is the phase where aeration and turning it becomes crucial. So that tends to be the longest bit, which at large scale can be weeks and weeks, or maybe even a couple of months. The quickest methods, if you follow kind of the Austrian science method for doing this, they reckon that you could produce finished compost in 6 to 7 weeks, which I find absolutely mind-blowing. But in order to do that, they're inoculating in the mesophilic stage, and then really, really monitoring, controlling, maybe turning twice a day, maybe watering during the thermophilic phase to keep it as active as possible, and to kind of move through that before moving on to the cooling phase, which is kind of carried out by the same organisms as at the start. So there's mesophilic, which are finishing off the decomposition and stabilizing the compost. Now, if you've added your colloidal solids, so at this point where it's really helpful to have that material, because those kind of microbes, those mesophilic microbes, are going to tuck all the nutrients that they've been collecting onto that colloidal matter. And then the compost will continue to cool down and enter this maturation phase, where it kind of all begins to stabilize, temperatures drop down, hopefully don't go away completely, but drop down to a steady temperature as the pH drops as well. And the longer you leave it, likely the lower the pH will drop. And that maturation is really quite crucial. The longer you can leave it, as I kind of mentioned before, generally so much the better. If you're having to store it, ideally, if your licensing permits, store on soil, but keep it covered. That way it'll be getting inoculated with all those kind of lovely microbes. So that's kind of the theory behind it. In reality, all sorts of things can happen. You can get double spikes. When you get a double kind of peak, generally it kind of happens because the mixture wasn't kind of homogenized well enough at the start, and so that then can delay the whole process. You keep finding pockets, and I suppose disaster would be when you've kind of come down to the cooling phase, you think it's maturing, and then it suddenly rockets in temperature, or you get a rain, kind of a rain shower, and moisture content goes up, and then that drives temperature up, and it kind of seemingly all comes back to life. So important to understand what's happening throughout this process to know kind of where it is on that journey, or else you could conceivably kind of build something, think that you're doing the right things, turn it at the right times, and then it seems to kind of completely stabilize in temperature and drop, but actually it could be that something's gone wrong and the process has stopped. So actively monitoring it is really quite key. So that's thermophilic, which I touched on, because even in the compost pile, these things are kind of happening. Adding something to that can help preserve more of the nutrients you're trying to retain. Adding soil and compost can help retain even more and add good microbes. But there are other types of composting as well. One I've already mentioned. You've got an aerated static pile. So that's a little bit like your kind of pallets in the garden type composting, but on a much bigger scale. The air is often forced in from the bottom of the pile, so that it keeps those kind of digesting microbes fueled with oxygen. You're not as reliant on turning it all the time. You've got in-vessel composting. Now that's often used to get rid of food waste, or I think it has been used to get rid of kind of livestock losses as well. Tends to be the sort of domain of more industrial, and that truly is a process of pasteurization as well. Incredibly high temperatures, often controlled pressure as well, and you end up with something quite sludgy, very different to the type of compost I think we're kind of focused on in this. You've then obviously got vermicomposting that we touched on, using worms to accelerate organic waste decomposition. Bokashi composting, which is more kind of like fermentation. And then one that we've been coming across more and more over the last year or so is the Johnson Sue composting, which is a no-turn aerated method as well. I see someone's hand's popped up, and it's a good time to ask a question. That is Chris.
Hi there.
Hiya.
So my question is about the thermophilic stage and the temperature. Is there like any problems if you let the temperature get too high, like up into the 80s?
Yeah, I mean, you can do all kinds of damage to the material you're trying to kind of get the most out of. You can literally burn through oxygen quicker and kill the process off. You get fire risks at that temperature as well. And if you are at that kind of temperature and compost is exposed to rain, it can push it that much more. And you know, seen it. It's what you don't want. Things suddenly start to smolder. And if you haven't been on site, or you're when that's gone on, or you're not trained, you're often left with a white kind of powder that looks like certain types of bacteria as well, which is not ideal. But yeah, things don't operate as effectively or efficiently at that kind of threshold. And as much as it is nice to hit as a ceiling for making sure that you are removing pathogens and killing off seeds, I think if you are actively monitoring your compost, there's no need to let it get quite so high.
So about 70 degrees is about as hot as you want it to get.
Yeah. Yeah. A lot of people will kind of use 65 as their ceiling, but I think you've probably got a little bit of wiggle room on that.
Okay, that's great. Thank you.
All right. Cool. Well, I'm going to talk a little bit about compost testing, and why you might want to consider it. And I think there's a couple of angles I mentioned already. If you are looking to kind of seriously level up your compost making, or even get the most out of your manure, understanding well, you can look at the nutrient concentrations of your manure straight away, just get an understanding of that. But the C and N ratio is a great place to start if you're looking to incorporate it as a feedstock for compost. Same with other materials, your greens and your browns. Understanding what those balances are before you build is a great way to kind of start with success in mind and build these things in the right places, in the right volumes to get the best possible mixture. But at the end of the process, it is important to assess the maturity of the compost. So, and also the quality, to make sure it's safe and good enough for your use. So could be looking at nutrient concentrations, pathogen presence, and microbial diversity. If picking up on that last, microbial diversity, it is often useful. So we look for we look at all the bacteria and fungi within a sample, but there might be indicators that are a sign that a compost is well mature, such as actinomycetes, which are or actinobacteria, which are an indicator that this has matured. It's gone through that kind of cooling phase and is in the maturation phase. So unless you have a microscope and a train, that could be quite difficult to look at, although not beyond the domain of anybody. Anyway, kind of brief interlude. This is kind of what we do. This is our fridge actually this afternoon. You can see we take so much soil that it actually breaks fridge shelves. So what I'll do is kind of talk you through the sort of work that we do that would look at the microbiological side of materials, and then I'll kind of show you the sort of output. So composting process, as I've just shown you, is a biological process, and we're blessed because we've got some classic scientific tools that we can use that allow us to look at what's going on and how much of certain microorganisms exist. So we take samples from lots of different clients from all over the place. They range from soil, they range from compost materials, compost teas and extracts, which we'll talk about in a bit, and even products as well. And they have to get to us as fresh as possible because they're living and breathing. So as soon as they arrive and we've recorded them, we'll put them in the fridge before we start the process of analysis. This was our oven this afternoon. So I mentioned earlier, soil moisture is crucial for biological activity. That means all the work that we do, all the analyses that we do, we break everything back down to a dry weight basis, which enables us to account for seasonal variations. It's also helpful to understand how much water is in the sample when we receive it, as often if there's too much, we do see kind of unbalances within the kind of communities of microbes that we're looking at as well. So this is kind of the process I described earlier, actually. The steel cores that you see, they're part of a different analysis where we're looking at in-field bulk densities for people's carbon assessments, but we'll take a fraction of compost, soil, even manure. We're doing some work on chicken manure for anaerobic digesters at the moment, and we need to dry it and record the difference in weight to work out what the moisture content is, and we can use that on our end analyses. We then take another sample from the bag, a very tiny sample. Actually, it's only about a gram. We mix it in a buffer solution, which kind of suspends all the bacteria and all the fungi kind of exactly as received. So it won't get any more active, but it won't get any less active. We've got kind of a footprint, if you like, for that particular sample at that point in time, and then from that we can do various analytical work. We'll do other extractions looking at things like nematodes. So we'll take a big mass of material and we'll put it in water for a few days, letting the nematodes kind of fall through the sieve, and then we can look at the different feeder types of nematodes that we've got. If you look at the top sieve, you can see that that compost material is particularly fungal happy. You see the mycelium on the top of it. Then we do a little bit of culturing as well. So this is classic agar plating, but we put different dilutions of compost or soil onto kind of rows of agar plates to then incubate for a few days more, and we look at the different types of protozoa. Protozoa, their main food source is bacteria. Protozoa are the main food source of earthworms. So where now we're starting to do lots of work for people interested in biodiversity, natural regeneration or nature restoration projects, where they might be doing kind of wildlife or ecology surveys, it's not kind of unfeasible to be able to link kind of mammal life back to worm populations, back to protozoa populations, back to bacteria populations, which I think is really quite cool. But all of these kind of samples and preparations then go through our light microscope. We do some work under the normal kind of wavelength, and then we do other work under a fluorescent wavelength. And this is one of Brona's samples. What you're looking at is fungi, and this is actually active fungi under a fluorescent stain, under a fluorescent light, fungi emerging from a spore. The reason we look under particular wavelength of light is because the dyes that we use react with kind of the biochemicals, if you like, given out by the organisms we're looking at. It causes a reaction, and then we can pick that up under a particular wavelength of light, and then we can actually account for what proportion of the population of all the fungi that we've looked at is alive and active, because it's quite possible to accumulate lots of bacteria or lots of fungi in your soil, be able to measure it through all sorts of systems that exist, but a little bit like kind of applying fertilizers or nutrients and looking at what's available in the soil. It's only the active fraction that is alive and functioning at the time that's able to kind of contribute in terms of beneficial processes such as nutrient cycling. So it's kind of important to understand, of all the bacteria, of all the fungi, how much of it is alive and working at this particular point in time. So that's kind of one thing. Now I'm going to flip about and try and show you a video which is much more exciting. Right. Let's see if I can do this. I'm going to stop sharing that, and then I'm going to end that. Talk amongst yourselves while I struggle with technology. Right, here we go, here we go. Got it. All right, there we go. Does that come up all right? One of the organisms I haven't talked about, primarily because it comes a lot later in the process, and we focus on kind of compost and manure and things, but it's still very important is mycorrhizal fungi, which we do look at as well. This, I just kind of had to show this because you can see, I presume my cursor's kind of coming out, you can see the root running through this. All this around the outside that kind of looks like the root, that's actually the mycorrhizal fungi, which covers the roots. This is an ectomycorrhizal fungi. It's a symbiotic, mutualistic relationship. Basically, there's kind of a fusion that happens hopefully early in the plant's life. You see it a lot in trees. It's a really great indicator for how well connected a plant is with a soil. So a healthy soil, you'll tend to see a good kind of amount of root colonized. But that's kind of one of the things that we do. But I said I was going to show you a video. Let's try this. There we go. It's a little bit grainy. So the particles that you can see are the soil kind of particles as bits of organic matter, and then you can see there goes a nematode. These things here are ciliates that I mentioned that swim around feeding on bacteria. Then we have, this is really exciting. If anybody knows what it is, you feel free to unmute yourself and shout it out.
I think I know what it is. Is it begin with T?
Yeah, it does.
Is it a tardy bear or something like that?
No, you've mixed. So it's often called, I think, a water bear. Soil biologists will say they're so cute. Like they're really cute. You can imagine kind of mad, crazy kind of people with tardigrade t-shirts, which is what it is. So he's a little bit higher up, but it's actually eating a nematode, which is, I mean, say it's unusual. I'm sure it happens all the time in soil, but we don't see things like this too often. And when you do, they kind of, they really do stick out. So it's, yeah, really quite exciting to see that.
Was that in my sample?
Sadly not. I'm afraid.
But I was getting very excited there.
Yeah. I think this might have been, which is much more nematode orientated. Again, that's a really great image because we're looking at often kind of people think you're just looking in X and Y kind of axes, but actually you're looking down through the Z axis in samples as well. So it's quite unusual that you get everything within the same focal plane, and you can see this. Just move you all out of the way. You can see the this particular nematode over here just beautifully, and nematologists would be able to kind of zoom right down into the actual species. They look at the anatomy. Sorry, it's kind of one of the ways, the position of the anatomy, the distances between things, and from that they can work out what particular type it is. This is a bacterial feeder. Again, I'm not going to talk too much about that because we're going to talk about it some more in a minute. Yeah, so that's the kind of thing that we do. Let me reshare. That worked a lot better that time. Hope you can see.
Simon, how much?
Yeah.
How big are those things we're looking at there?
Oh, they are tiny. So probably that would have been, the tardigrade was probably 200 times magnification. Most of the work that we do is actually, relatively speaking, quite low, or people are surprised. They think that everything you do is at 1,000 times.
Yeah.
The bacterial work is a lot of it at 1,000 times. So for that, we need to put a drop of oil between the objective on the microscope and the top of the slide that we're looking at, so that it removes the air void. But almost everything else is 100 times or 200 times.
That's amazing. That's pretty cool to see.
Yeah, it definitely is, and it's we kind of always see new and interesting things. Right, let's bring that down here. Yeah, let me just stop that again. I'm gonna confuse myself with screens. That's what happens when you kind of go off piste with images, I'm afraid. There we go. There. Right, back in the room. Cool. Yeah, so it's quite a complex process. That produces a report like this. So this Brona, this is your 4-month-old vermicast.
Okay.
It's, I have to say, and I'm not just saying this, really quite good. The things that jump out to me, and this is kind of a recurring theme of the moment with lots of people I talk to, moisture content. I've said it about 100 times already, but we measure the moisture content. So the sample that you sent to us, 61% of its weight was water. That's incredibly important. And remember, I kind of talked about the optimum composting temperature ranges, 40 to 60% as well. It's kind of no coincidence, because earthworms, one of the attractions of this particular type of composting is they do a lot of the hard work for you and homogenize things quite nicely. But when you've got a good moisture content, things tend to be more active. Now, I just spoke about kind of total levels of things, total bacteria, total fungi, and the proportion that's active. So what's quite nice about this is, for a vermicompost, it's actually got quite a high level of fungi. So this is total fungi. That means the active and inactive fungi. We're not looking at spores, so this is referring to hyphal strands. 63, and this is micrograms, so that's 10 to the minus of 6. It's 10 to the 63 times 10 to the minus 6 kind of mass of fungi per gram of vermicast out of a total of 557. That's quite a good proportion. Generally, we find vermicompost to be more bacterial, but that's to be expected quite often because, as I mentioned, earthworms tend to eat a lot of protozoa as well. I know these are composting worms, but protozoas feature quite heavily within the diet. Their main source of food is bacteria. So you tend to end up with slightly higher bacterial populations, but as it happens, is quite nicely balanced. I'm not going to talk too much about organism ratios, but the protozoa levels are crazy. I think this is actually, there's over 1 million flagellates per gram, which is, I can't remember seeing a number that high for a while. Then you've got amoeba. That's quite a nice population, although it's, ideally we'd have more, and then we can kind of see it's reflected. Ideally, these three would kind of be in better proportions. However, I think a lot of that is just driven by the very high bacteria numbers. Dependent on conditions, one of the types always tends to kind of dominate slightly more. The nematode numbers are really good. Again, we look at the feeder types. So that's to say whether their primary food source is fungi or bacteria, if they eat each other, if they eat plants, they're the sorts which you don't really want to be too dominant. kind of your root knot gall nematodes and things like that, potato cyst nematodes. Of the type that we saw, all of them are bacterial feeders, which again kind of is indicative it's quite a bacterial dominant compost that you've got there. And then we've got a value based on the numbers of protozoa, the numbers of bacteria. What this is, it's kind of a calculation for the biological nitrogen potential. So if this vermicast was exactly the same, and it was kind of applied across a hectare to 10 centimeters depth, I think it is, then you could generate the equivalent of 336 kilograms of nitrogen through natural nutrient cycling over a 3-month or 3 to 6 month period, which is kind of at the top range. It just basically means it's very active. The nutrient cycling kind of characteristic is incredibly high.
That's very surprising. Potential for nitrogen cycling. What would be an application rate? Is that thinking about it as an extract going out as a soil drench or as a pure
as a straight amendment? So this is, we'll talk about kind of extracts and things in a minute, but this relates to the material as is. So if you were to, and I'm sure it is based on a 10 centimeter depth, so you'd need an incredible high volume. I might be wrong on that, but it has a high natural nitrogen cycling capability. Normally, this number actually relates. It's perhaps more useful sometimes from a soil perspective, because I'd need to look at and calculate from bulk density how much you would need to generate that, how much you would need to apply, sorry. And I know with vermicompost, often for the effort, the actual volume you get is quite low or slow unless you've got a massive scale. But I suppose other things, if you wanted to kind of take this up a level, you'd kind of think, right, now we've got this finished material, I want to increase the fungi levels even more. Maybe kind of looking at leaving it longer, or leaving it longer in a certain location, like I said, under trees, or perhaps adding something to it that contains more fungi, blending it with something to try and drive those numbers up. But all in all, it was very good.
Cool.
Yeah.
I'm surprised for it old as well. So
you were surprised what, sorry?
Because it was old, it was just sitting
in
the bag.
Yeah. Right.
Simon, so a quick question about that. If you had more wood chips as a feed source, would that increase the fungal activity, or is that just sort of doesn't really matter?
I'm sure it matters tremendously, because there'll be indigenous fungi within the wood chips themselves, and I imagine a proportion would be preserved even after passing through a worm. But I suppose it's getting them into a form that is, in this particular case, vermicast, getting them in a form that's kind of digestible. And I think, leaf mold was one of the other samples that Brona sent. I can't remember that, but often people will include leaf mold as a food for worms to try and elevate those numbers as well.
Cool.
Yeah. Okay. Let's just move you all so that you can see. So we're just going to spend the last kind of few minutes talking about amendments, as a it's kind of informed that was of interest. Let me just see if I can move that, as it appears to have frozen. Can anyone spot what's wrong with this picture? Feel free to, go back. And maybe it's not wrong, but it's perhaps a slightly incorrect statement. Basically, it relates to the misunderstanding, perhaps, or the confusion between what an extract is and what compost tea is. They're both concentrated solutions that tend to be made from compost or manure to enhance soil fertility. You can make all sorts of other teas. I think of comfrey tea. I remember making some of that last year. That was very smelly. Nettle tea. And they, again, I'm lucky I've got microscopes and I can look at them. They definitely produce lots of microorganisms, and I'm sure from the smell that they definitely provide fertility benefits. But when you're making an extract out of compost or manures, you are getting the biology that's attached with that. And that's great because it means that you can kind of introduce that biology to your growing environment, or your greenhouse, your pot plants, your house plants, your vines, your trees, your fields, your grasses, whatever. So, but this picture's slightly wrong in so much as it says making compost tea. I suppose aerated compost tea might be a bit more appropriate. This could be considered an extract. I think this is actually kind of a biodynamic process. So you use a barrel, you stir in a figure of 8, which I've done as well. It's really good fun, but that is more kind of extract. So there we go. So what is an extract? Well, this is kind of perhaps a slightly better picture, although maybe you wouldn't necessarily submerge your hands all the time. You're steeping your material in water for a period of time. Sometimes that's quite a short space of time, like you hear people making a microbial extract. So they will literally put a load of compost in a bucket with water, dechlorinated water, and then they'll get one of those kind of plasterers whisks on a drill, zhuzh it around in the bucket, and what happens is all the microbes that are attached to the particles of compost then go up into suspension in the water. You siphon off that water fairly quickly after doing that, and then you go and apply them to your crops or plants or soils. You can use kind of straining bags like this. I wouldn't necessarily recommend using your hands unless you've kind of rubbed them in compost first, because our hands have all kinds of oils, and if you've washed them with soap, those can be antibacterial. But yeah, that's so an extract. When I think of an extract, it's about solubilizing your material to put all those microbes and maybe some of the kind of soluble nutrients into liquid in a suspension over a quite short period of time. Compared with compost tea, this is an old picture of my son from about, oh goodness me, 10 years ago, I should imagine, with one of my first brewers, which you can see he is doing a good job underneath a electrical cable. We won't talk about health and safety there, but this thing is bubbling away, which is indicative of microbial growth. And that's happening because rather than just stir the material in our dechlorinated water, we're actually introducing other stimulants. So we're in this particular case, actually, I was using humic acid and molasses. And what that is doing is providing a really readily accessible source of food and energy for all the bacteria primarily, but the fungi too, to eat it, grow and reproduce. As they're reproducing really, really quickly, they're consuming more oxygen. So we need to have a continuous supply of oxygen in our tank, which is while the bubbles are forming as well. And they'll continue to eat that over a period of, say, 1 day. You hear some people might want to brew their compost tea for 2 days. And then, just as with the extract, at the end of that period, you take the liquid. If you're going to spray it, you often siphon it so that it runs through your kind of hose or spray lines, but you could apply it kind of unstrained through the spout of the watering can, or strain it backpack sprayer. But fundamentally, the difference here is you're not just taking the microbes attached to the material, you are increasing their numbers, the concentrations of them. You're culturing effectively over a period of time. Different food sources will promote different microbes. Different temperatures will accelerate that rate of growth, and there's a lot of contention over, if you add a heater, are all those microbes that you've grown at a warmer temperature going to die when you apply them to cold soil? I think the answer is probably a lot of them will, yes, but a lot will survive. How many? We don't know unless we test. And yeah, the timeframes over which you're culturing it, the longer you brew tends to be the more fungal your mixture will end up going. But in either case, the really important factor here is that the starting material, whether it's an extract or whether it's a compost tea, the starting material, your compost, your manure, the profile of that at the beginning is kind of retained. It's just that with the compost tea, you've magnified it somewhat, and even if you've tried to kind of shape it to be more fungal dominant, if that's what you're looking for, you need a more fungal dominant starting material. So unlike the extract where you've got kind of a kind of nutrient fix from the material and the attached microbes, with the compost tea you've got that nutrient boost. It's unfortunately a bit slower to make and a bit more kind of hands-on, I suppose. The clean down is a nightmare. You have to kind of operate with quite high standards for hygiene to not get contamination. But the main difference is it increases the concentrations of microbes, and if you do this correctly, you can boost soil kind of micro communities. That's the brewer setup I've got now, which is slightly different, a bit more evolved. This is kind of my own pleasure, I suppose. It is imperfect. As always, there's modifications I would rather make to get it to kind of work better, but the shape of the tank, the piping, the injection of air induces a vortex within it. So kind of material is continuously falling and being reoxygenated. I can get some really nice kind of brews as a consequence of it. But there's all sorts of ways that you can brew. I mean, the vortex brew is kind of one of the more complex. You've got classic kind of aeration where you can use a diaphragm, or if you're starting out, even an air stone that you've got to kind of really clean so that you don't get biofilms that can be harmful. You've got lifting compost tea brewers and all sorts. Yeah, I'd really like to do a practical workshop on compost tea at some point, but it's difficult to do over computers, I'm afraid. When you've got your mixture, there's a whole heap of ways that you can go and apply it. Straight from a watering can is often the easiest, but it's not much help if you've got 100 acres. I've heard of people that have made really high quality compost using that Johnson Sue method I spoke about earlier, which they've done in an IBC. So taking quite a, relatively speaking, quite a small amount of compost, brewing with it, so amplifying the amount of microbes, and then they've put it on the back of their drilling machine with a bit of modification, and they put a squirt behind the seed in the furrow as they've gone out and drilled. And I'm hearing increasingly more people look to do that. The classic that from the Soil Association trial that I mentioned earlier, The way that most people probably are still using compost tea is going out and spraying. So kind of just straight boom spraying. I remember standing in a incredibly hot, dry field in Dorset watching a 36-meter boom off the back of a tractor spraying compost tea and thinking this is just not going to work because it is bone dry. I think the crops need water more than they need microbes at the moment. So yeah, spraying it's kind of the classic. What you're looking at here is a lovely oak tree in Burley House in Stamfordshire, where we actually, it's not an oak tree, I think it's actually a plane tree by the looks of it, but we're treating declining tree health with a combination of wood chip mulch and compost teas to the soil and foliar. If you're spraying, it's important to just kind of really think about the setup, because you're dealing with little life forms. It's important to not kind of harm them in the process of transferring them. So there's a whole kind of series of things to consider, from pump types and pressure ratings and things like that when you are going out and spraying, which I'm happy to speak to anyone about offline if they want to know more. But you'll be pleased to know that we're kind of at the end. I guess there's two key takeaways from all of this. I mean, there's hopefully lots of little points that I've covered, but the key kind of starting point is that compost and farmyard manure management is an active and dynamic process. It's this idea that it's not just a recipe that you set and forget. Even if you follow the instructions, you need to check on it and alter things, because nature is imperfect. So keeping records is incredibly important. Using your senses, sight, smell, touch, you know, the squeeze test, really important in the absence of any equipment. I mean, if you don't know how to use the equipment, it's pointless anyway. But if something looks wrong, smells wrong, it's probably wrong. And if it smells wrong, it's likely that you're probably going to need to either modify the moisture content or carbon nitrogen balance through the addition of bulking materials. So that's the first thing. Active management is key. And the second really kind of ties with work we do in the lab, which is try and measure everything, because if you can measure it, you can manage it. And I've nicked that from someone. I can't remember his name. It's a very famous quote, isn't it? Kind of try and get as much of an understanding. If you're kind of looking to get the most out of your compost, look at what's going into your compost and try and optimize it at that stage. From what I've learned about composting, what I've seen from others, from what I've been shown, often all the fancy amendments and products that people kind of recommend to add to your compost, 9 times out of 10, they're better placed being included at the start of the compost process, so that they kind of worked through that natural kind of digestion microbial process, and then assigned in the right places, like with the the addition of compost and soil, that you'll end up with something kind of much more high quality, much more consistent in that way. But I think there's probably a bit of apprehension sometimes because of the cost of some of the addition of these products. But we can trust in nature perhaps a little bit more. But in any event, take a record, test things. If you understand what you've got, you can make changes and do something about it, and then measure those changes and take further action. So there you go.
Thanks, Simon. Yeah, we'll let anyone jump in. Chris, did you want to ask something?
Yes, I was going to ask earlier on, you were talking about, I think it was the colloids, which is like adding soil to the compost pile. Is there a certain ratio that we should try and follow? I'm currently adding like a bit of soil at the start of the composting period, and it's probably like at a ratio of 1 part soil to 10 parts other ingredients.
Wow. Well, hats off to you, because that's awesome. A compost as well, you can put some of the finished product back into it. There isn't, unfortunately, like an absolute defined amount that I've come across. I've read in the Austrian method different rates to try, which might be more helpful, but they're percentages. So whenever you add anything to a compost to enhance it, it tends to be the 1, 3, 5, or 7%. A lot of the time, around 3% is enough, and that's by volume.
Okay, so 3% soil might be enough. I'm probably adding a lot more than that.
Yeah.
No, in fact, it's more like 10% probably.
Yeah. It's, yeah, I mean, it's a bit of an odd thing that they're odd low numbers. I guess it's kind of like, you know, the Fibonacci sequence. Why does it work? It just works.
Yeah.
But it tends to be quite a low amount and an odd number, 3 or 5%. Yeah. I'd probably add, so I make like a static pile, like a turn, like pallets, and I'd probably add no more than 5%. And it does pull through. You notice, the more you add, the more soil-like it ends. But I do notice that the smell changes, but the microbial concentrations also change, and I use it for brewing a lot of the time.
I see Connor, I think, had his hand up next there.
Thanks, Bruno. Simon, how important is diversity of feedstock for quality finished compost?
Is what, sorry, the feedstock?
The diversity of the feedstock.
Yeah, that's a good question. Like, absolutely. The more different types of material you can include, the better. I mean, quantify it again, it's kind of hard, but ultimately, the more diverse in, the more diverse out, the more you can get as is reasonably practical, so much the better. And appreciate in some instances that's quite difficult, where you've got large volumes of one particular type of material. It's kind of being as inventive as you can to incorporate more different types of materials, even though you've got that kind of hot process in the composting. It doesn't wipe out all the good bacteria and fungi that we want at the end of it. They live still. So the more different types that you can bring in, ultimately they're going to end up in your soil, and you have a more diverse, resilient soil at the end of it.
Nice one. Thank
you. Esther, you had a question?
Thanks, Simon, for a really interesting webinar. I think one of the questions we have, we're a sheep farm, but we are also, most of our manure is actually coming from horses as well.
Okay.
Is there anything specifically you would recommend around horse manure? And also, we collect a lot of our horse manure off of their winter sand paddocks, so they're in an all weather. So we're collecting it, so it's very manure heavy. And then some of it will be combined with shavings, and then the straw out of the lambing shed. What would any specific considerations around that you would recommend we think about?
I suppose considerations would be, and I'm actually thinking not of horses, but of the chickens I mentioned as animals and similar processes. What we've found is vet treatments that the animals have gone through have an impact, whether it's the inclusion of certain medicines. So, okay, if the horses have been wormed, that's not great because that will come through in their poo and that will go through in your compost. It might be unavoidable. I'm not saying it's a calamity, but it's something to be mindful of, because then if you're looking at the health of your soil that this has been applied to, it might have implications in terms of the nematode populations, and we would expect to see that in kind of unbalanced numbers, perhaps, more one type, less of another. So that's some maybe questions to ask. Otherwise, perhaps going back to the sort of the nutrient concentration question earlier, I'd probably be mindful of that because they're probably on a fairly monotonous diet, I imagine, the horses, or supplemented diet that could also impact the quality of the manure. So, yeah, rather than just look at the things to add to it, I'd maybe be inclined to understand a bit more about what might be in the manure.
Thank you, Simon. Simon, just to follow on from something you said there, and apologies if I missed it because I was working. Looking at the nematodes and the worming, surely that's a consideration across the board for everybody. If we are worming, doing some batch worming, I know we are all moving to sensible worming, but is there something we should be considering there more when it comes to manure and compost?
Yeah, I mean, absolutely. It's something everybody should be aware of. We did some microscope training a couple of weeks back, and teaching people how to do qualitative analysis. It's like a slightly different form of what we do in the laboratory. But one of the advantages of that is people being able to do their own fecal egg counts, and choose to not worm animals unless they absolutely need it. But sometimes you need to, and this kind of comes back to there's the optimum, there's the perfect, and then there's the real world, and you have to get on. And I suppose it's not really answering your question, but yes, everybody should have the same consideration, and it's just being mindful of this is going on. Maybe have a look at your soil, or have a look at your compost or your manure, and just be aware that if it's peaking, you might need to mitigate for that a little bit more in the future. Or if you're not, that might just send you on a particular path.
Thank you very much. Thank you.
Esther, was it you that we were chatting about this before? If, say, we wormed sheep and we moved them onto a paddock after worming, and we kind of treated that like the sacrificial paddock until it was maybe out of their system, and then we're trying to think about what would the treatment we would do to that paddock to try and mitigate the damage of the wormer. Because I can see with the horses.
Yeah, no, I don't think it was me had that specific conversation with you, but I suppose from a perspective of what it does damage. We know that the anthelmintics are damaging a lot of things, and so that's why it's so important to have appropriate worming care with your fecal egg counts and your salivary tests and all like that. But one of the things I think to pick up on there is just a bit of veterinary advice from the horse vet is don't move them until they've had a couple of days. Don't worm and move. Keep them where they're at, and then they don't come back onto that field for quite a period of time. But horses, interestingly, everybody's talking about poo picking and lifting the feces out of the field, which is so contrary to everything we talk about in the other species, and that's a very different subject, but quite interesting as well.
Yeah.
Very good. Anyone else have any questions? You might not have put your hands up, but jump in. No. there was something I wanted to ask you about, Simon, there when we were talking about carbon to nitrogen ratios. Do you measure that in soil a lot and see quite varying results? It's something that we've been chatting about. Isn't it a unique scenario we have in Northern Ireland because we have so much organic manure to put out?
Yeah.
How we driven our soils quite either nitrogen rich or carbon rich, and have we skewed that balance, and it's part of a compaction issue and nutrient lockup? And it'd be interesting to hear your thoughts on that.
Yeah, I mean, I think we don't see many soils. In fact, I can't think of any that I've measured at 24:1. I've actually seen contrary advice from other laboratories that say if it's above, I think if it gets above 12:1, that's bad. So I don't think it's particularly well understood, but I also think that often the timeframes that we're looking at are particularly skewed as well. We kind of think very short term, and ultimately, given a long enough period of time, things will kind of revert to back to natural equilibrium. So any interventions we make will probably have quite a significant effect, but then for it to kind of restore back to something more more balanced is beyond our kind of comprehension, and we'd invariably have to kind of do something with it. So yeah, most of the soils that we see will have a low carbon nitrogen ratio.
Okay, would you say that's soils that their organic matter is measuring quite low as well in relation to that? Yeah,
very often. Yeah,
I think that would be quite interesting to see the difference of some of our soils, because our organic matter is quite high here in a lot of farms. And we're talking about making compost into getting it into that right carbon to nitrogen ratio, and then we go and put it out in the soils, and then they're not in the right kind of balance of what is a lot of your work getting wasted?
Highly possibly. I mean, you kind of think especially permanent pastures, which to all intents microbially are really quite healthy, and they're just kind of driving themselves and driving their own organic matter through fine root turnover. So I think kind of looking at different contexts would be really interesting, as well as looking at the individual organic matter levels, as well as looking at the types of organic matter that's applied. And I think organic matter quality is that's kind of like a bit of a soapbox subject for me, because kind of before the whole move to regenerative agriculture, everybody was kind of soil health focused. Then it became very soil organic matter focused before soil carbon kind of became the new thing. And no one really gave, I don't think, soil organic matter a chance. The opinions on it range from I chop my straw and that's sufficient, I don't need to do anything else, to I don't need to compost because I've got walking compost machines. But that word someone mentioned earlier about diversity, it's if you're always feeding the same meal to the same things over and over, at some point, it just doesn't feel right long term. Like, variety is the spice of life. More different types of organic matter. It's kind of why that question about feedstocks for compost, the more different types of materials you can incorporate, the more diversity you get out there, the higher the quality, the more different types of food that appeal to the wider range of microbes in the soil, you'll stimulate more. So yeah, I think organic matter quality context is key as well. Well,
we have to ask now what you asked there what animal was producing the most.
Oh yes, yeah. I wrote it down so I wouldn't forget as well. Pigeon.
Oh no way!
Pigeon poo.
It might have been rabbit because it worked brilliantly in the vermicompost.
Did it?
Pigeons. Okay, well, I'm not about to start wanting to attract loads of pigeons.
Pigeon farming, yeah.
Very good. Well, there's people in London can scrape it off the streets and
exactly. Leave it to the city folk, yeah.
Yeah. I think it's been really interesting. And we'd love to hope you're still up for coming over to Feels Good. And just to let you know, that's happening on the 6th of September this year.
Cool.
It's up on Eventbrite now for early bird tickets. And Simon, we'll have to talk about how we can get your vortex extraction machine maybe over. Do some mini compost piles or something. But it'd be great to get some demos with you for the festival and let people see firsthand.
Yeah, that'd be great. I'd love to do that. Yeah, have lots of different types of brewers, maybe kind of a build a brewer face-off challenge or something. Yeah.
That'd be great. Yeah. Oh, thanks so much.
Super. All right.
And you give us so much time as well. Super. Right. We'll have another catch up soon. I'm dying to see the rest of my results.
Yeah. Super. All right. Thanks very much.
Thank you.
Thanks, everyone.
Thank you, Simon.
Cheers. Bye.
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 Thank you very much.
Thank you. Bye. Bye.
Cheers, everyone.